big hello to my wonderful PU2 champions. I hope you're doing absolutely great. Well, today we are going to go ahead with the chapter Molecular Basis of Inheritance, one of the most important chapters. Why important? Because, my dear children, it gives you 13 marks in the examination hall. 10 marks come from the 5-marker session itself because 2 questions are there in 5 markers from this chapter, 1 2-marker, and there will be 1 1-marker question. Now, my dear children, one very important thing that I want to tell you is, after watching this video, I'm pretty sure you will grab these 13 marks. Pretty sure. Why? Because I've taken from this year questions from this year's model question paper, from the previous year questions, from the revised question bank. Every question which might be difficult for you or every concepts which are usually being repeated are covered over here. So you need not worry about any single thing. You just need to listen to me. Do not make your notes. After, okay, well, let's have a challenge. Let's just focus on straight 30 minutes, nothing else. No focus on nothing else out there. Just in 30 minutes, you and me, I talk, you listen to me carefully. After 30 minutes, pull out a pen and a paper and write what is important. Whatever important concepts I am telling, please write them in your way. Short way, but in one particular page. So in one particular page, all your important concepts should be there. This can be really important for you during revision. This page can be referred. Okay? So let's go ahead with the first part, that is the model question paper questions of the year 2025-26. And over here, I have the first question, which is model question paper 1 question number 4. Another chemical name for the nitrogenous base thiamine is. What is the other name for thiamine? What is the other name for thiamine? So, my dear children, we have 2 types of nitrogenous bases. One is purines, another one is pyrimidines. Purines, it is A as well as it is G. It is adenine and guanine. Pyrimidine, you have thiamine as well as cytosine. This is the case of DNA, my dear children. Adenine base pairs with thiamine and guanine base pairs with cytosine. But in the case of RNA, which was actually the first genetic material, okay, you did not have thiamine. Instead of thiamine, you had uracil. But why do you call RNA as the first genetic material? Because something better came behind it, and that was the DNA. There were some changes in the RNA which later helped in the formation of what? DNA. And what was the change? My dear children, uracil was less stable. Uracil is a nitrogenous base which is less stable. Why not do some changes in uracil? On the 5th carbon, let's add a methyl group. Let's add a methyl group, and that's how you got thiamine, another nitrogenous base which you find in the DNA only. So uracil in RNA was later converted to what? Thiamine. Okay, it happened and it was good. So my dear children, this was being taken up. So over here we have the answer as 5-methyl uracil, not 2-methyl uracil, not 3-methyl uracil, not even 4-methyl uracil. It's just 5-methyl uracil. Over here you can see there's a methyl group being added, CH3 group being added, whereas a normal uracil you do not have it. Now the next question that we have in MQP1 is 2-marker question, and this is question number 24. Name the methodologies used in human genome sequencing. Human genome as in our genetic, okay, our DNA sequencing. What are the methods that were used? So there were 2 methods being used. One is expressed sequence tags. That is, you know, we have our DNA. In our DNA, there are regions that codes for protein, you call them exons, and regions that do not code for protein, you call them as introns. You make the DNA undergo transcription, and that's how you get RNA. You call this RNA as hnRNA. So whatever information is there in the RNA format, it is being transferred up over here. Exons are still here, introns are also still here. Now, you do not need introns because they do not code for anything. Are they coding for anything? No, they're just in between the exons. So why not just remove it through processing, and why not have an RNA which just have the exons? That means the one that is going to make the proteins. What do you call this as? You call this strand as now mRNA. You call this as mRNA. So in expressed sequence tag, only the genes expressed as RNA were identified and sequenced. So only the part of the genes which were basically coding for something, those were identified and those were sequenced. That means introns were not sequenced. you have something incompletely done. You have only the exons being sequenced. Now there is whole genome sequencing. What do you do over here? The entire genome, whether it's coding sequence, whether it's non-coding sequence, all of them were sequenced. And then later on their functions were assigned. Sequencing was done, later on what does it do, what does it do, what does it do was being assigned. So express sequence tag is one, and whole genome sequencing, or you also call it sequence annotation, is the another method that we have. Cool. Now, after the 2 marker, we have a 3 marker question. This is question number 37. DNA is genetic material in both prokaryotes as well as eukaryotes. Yet the packaging process in their cells is not the same. Explain. So is DNA the genetic material in both prokaryotes and eukaryotes? What are prokaryotes? Ma'am, bacteria is a prokaryote. Prokaryotes are the ones which have simpler cells. They do not have a proper, what do you call, nuclear membrane to separate the nucleus and the cytoplasm and all. Their genetic material is just lying around, my dear children. It's just lying around. Everything happens within the cell without any boundaries. They do not have membrane-bound cell organelles and all things like that. Okay, whereas in the case of eukaryotes, things are not pretty much like that. You have a nucleus with a nuclear membrane. Inside the nucleus, this DNA has to fit in. So obviously, the packaging process would also be different. So let's read it out and then I'll explain. So DNA is a genetic material in both prokaryotes and eukaryotes, but its packaging differs due to differences in the genome size and the cell organization. 2 things. In the bacteria, the DNA size, my dear children, the genome size is very less. In comparison with what eukaryotic genome size. We have 6.6 into 10 to the power 9 base pairs, my dear children. Where will you fit it in? Your nucleus is so small. It's very difficult, right? So in prokaryotes, so 2 differences are there, genome size and also the cell organization. So cell organization, I already told you, it's having cell membranes and, it has nuclear membrane and stuff like that. But genome size is also one thing. In the case of prokaryotes, they have less. In the case of us, we have more. In prokaryotes, DNA is naked, circular and present in the nucleoid region without histones because of the small genome and absence of a nucleus. So they do not have a nucleus, so why to be worried? Suppose this is the place which is there. So in this particular place itself, you have everything. You would be having everything. Your genetic material is randomly out there, and the size is also not too much. Genome size is also not too much, and the cellular structure is also not like that you have clear differentiation between things. Now in the case of eukaryotes, things are different. In the case of eukaryotes, my dear children, we have a cell. Inside the cell nucleus, here within it, the DNA has to fit in, and our DNA is bigger also. So we need help of some very important proteins, and those proteins are mentioned over here. It's known as histone octamer. So my dear children, I'll tell you. 1 histone protein, 2 histone protein, 3, 4, 5, 6, 7, 8. 8 histone proteins comes together. You will now call this as histone octamer. Our DNA which is there, it will wrap around this histone protein. How many times? It will wrap around the histone protein twice. 2 times. It will wrap around twice. How many DNA, what do you call, nitrogenous bases will be there, or how many nitrogenous bases or nucleotides will be there, my dear children? Almost 200. Almost 200 base pairs would be there in this, what do you call, in this turn that you see. So twice it turns, so that means 200 base pairs would be right up over here. Why do you think that the DNA wraps around the histone? DNA is negatively charged component. Is there something else in the case of histone? Because if both of them are negative, they will repel. So histone, my dear children, histone protein is made majorly of 2 amino acids. One is lysine and the other one is arginine. You have 2 of these amino acids which are positively charged, and that attracts the negatively charged DNA to wrap around them. So this whole thing that you see is now known as what? A nucleosome. It is now known as a nucleosome. And there will be one more histone protein outside. You will call this as H1 histone protein, as you can see over here. So it's a histone octamer, and you have the DNA wrapped around twice. Almost 200 base pairs of DNA would be right up over here. So in eukaryotes, DNA is associated with histones. About 200 base pairs of DNA wraps around a histone octamer to form a nucleosome. These nucleosomes coil further to form what? Chromatin. Now the nucleosomes which are there, they will come closer, come closer, come closer. What you will be having? You will be having something like this. they will come closer to help in the formation of what? Help in the formation of something that you call as chromatin. And now this chromatin again finally condenses to form what? To form a structure which you call as chromosome. So you have to make it such compact structure to fit this whole genome inside the nucleus. So that is why the packaging process is entirely different, and here I explained it to you as well. Please make sure you make the diagram of nucleosome, super important. Okay, works. So difference lies in genomic size as well as cellular organization. Now did I miss some question out there? Because one more question should be there, right? Okay, we'll see through it. Now, if an mRNA, this is MQP2 question number 4. If an mRNA having this thing undergoes translation, how many types of amino acids would be present? If an mRNA having a sequence like this, how many types of amino acids would be present? So ma'am, we have studied that in that 3 codons would be coming together to form amino acid. So AUD stands for methionine, UU phenylalanine, so methionine, then phenylalanine, again phenylalanine, and this is stop codon. So basically phenylalanine is thrice, but I would not count it as thrice. One methionine, they are asking you how many types of amino acids. So 2 types, one methionine and one phenylalanine. Do you think that's the answer? Do you think that's the answer? Because anyway UAA is a stop codon, right? So what do you think? Is 2 types the answer for this question? If at all you think that's the answer, my dear children, you're getting things wrong. Please understand, the mRNA should be ulta, should be read like this. 5' to 3' direction. It should be in 5' to 3' direction. So in that case, this is one codon, this is another codon. Now the same thing is repeated, and now this would be the third codon, GUA. It is read like that. So 3 types of amino acids are going to be formed. If at all you see, if you make the ulta, do not transfer anything up, just write the sequence A, like that. Okay, in that case you have 3 of them. One is this, another is this, it's just repeated again, so you do not count it every time, and then this is another amino acid. 3 types of amino acids are going to be formed. Okay, yes. Next question, draw a label diagram of the nucleosome. So we just have to draw this. I explained you one more time. If you want, I can explain. We have 1, 2, 3, 4 histone proteins, and my dear children, at the back side also try to make. Now wrap around the DNA. It should be wrapping around twice, and it should be coming up over here where you have another histone protein which you call as what? H1 histone. Mark them properly and you have got your answer. That's all. Okay, mark them properly that this is DNA, this is what? Histone. So I did not make it properly, thinking that the picture is almost there. They can make properly, it's not like I do not know. So you might think that ma'am is not making, so I will also not make. So don't do like that, which is why I am making right now. So our DNA had okay, and now we have our histone protein right over here. You call this histone protein as what? H1 histone. This is your another histone protein. All of them together you form the octamer. This is your DNA, and I think that's it. That would be more than enough. Okay, so please make it properly, super important, and the whole structure that you see from here to here is known as nucleosome. Cool. Now explain Griffith's experiment, transforming principle experiment. This is MQP2 question number 38. What was Griffith's experiment, my dear children? It's a 5-marker question and it usually comes up. So Griffith's experiment is basically, he was trying to find what is the genetic material, and he was working on a bacteria whose name is Streptococcus pneumoniae. Okay, and this bacteria had 2 strains of it. One is R strain or you call it rough strain, and one is S strain or you call it smooth strain. Why there's S strain or smooth strain? Because it has an extra covering right up over here, okay, which is made up of a polysaccharide. This covering is made up of what? A polysaccharide, which makes it smooth. But you know what? Apart from making it smooth, it also makes it virulent. That means if an organism is infected with S strain, they will be dying. That is what you mean by being virulent. Whereas R strain did not have this particular membrane around it, polysaccharide membrane around it, which is why it was rough, and it is considered to be non-virulent. Non-virulent. Now what did you do? You took some mice. Griffith actually took a mouse. So over here, you can clearly see the first thing that he did that, he took a mice and he put the R strain into the mice. R strain is the rough strain. Yeah, it does not cause any trouble. The mice lives. Then he put live S strain. Now when S strain has been inserted into the mice, what happened? The mouse basically died. Now he saw that this is what we studied also, virulent, non-virulent. Now he did one more thing. He heat killed the S strain. That means he treated the S strain with some heat. And when he did that, basically some changes happened up over here. Some changes happened up over here. In the heat killed S strain, my dear children, when this was being put out, the mice lived. The mouse which was there, it lived. Why? Because probably something was getting changed in the S strain. Something was being changed because of the effect of heat, and which is exactly why it was not able to show its virulent effect, and that is exactly why the mouse lived. So he thought, okay, so by mixing this, the mouse is living. By putting this also, the mouse is living. Why not mix both of them? Why not mix both of them? So he did the next treatment where heat killed S strain and live R strain were mixed. Both of them were not causing any trouble until now. But now the mouse died. He was like, what is happening? So he got to a conclusion that the R strain which is there, it is getting transformed. It is getting transformed into the S strain, into the live S strain, not the heat killed S strain, the live S strain. Why did he come to a conclusion like that? Because he could find the live S strain in the mouse. We didn't put the live S strain. How did it come? So probably something was transforming the R strain into the S strain, because live S strain was recovered from this group of mice. Then he was like, what could be that thing? And basically whatever is that thing is responsible for showing the virulent behavior. So he called that thing as the transforming principle. He called that thing as transforming principle. That transforming principle is getting transferred from heat killed S strain to R strain, but he was not able to deduce what exactly is it. Is it a DNA? Is it RNA? Is it protein? Nothing. So this was Griffith's transforming principle experiment. Later, MacLeod, McCarthy, Avery, all of them, they came together and they started doing their experiment. I'll tell you what happened in their experiment. So basically, they took the same thing. Okay, they took the same thing. Over here, in the heat-killed S strain, they made sure they treated it with RNAs first. They treated it with RNAs so that any RNA in the heat-killed S strain would be killed, just the heat-killed S strain, okay? And then they mixed it with the live R strain and gave it to the mouse. They found that still the mouse was dying, because only the RNA is getting what? Hampered. The RNA is getting hampered. Still there is something that is responsible for transformation, because no change is happening up over here when I'm treating with RNAs. Why not treat it with something else that can kill proteins? Because proteins are my next doubt, right? So I will digest the protein with another enzyme, that is protease. So protease plus heat killed S strain put into live R strain, still transformation was happening. No changes. Even though proteins were not there, still transformation is happening. Still the mouse is dying. So this is also not the one. Then finally they want to make sure whether DNA is the one. So they treated it with DNAs. When DNAs was being put down, the DNA of the S strain is being gone. And now, my dear children, they saw that the mouse was living. The mouse was living. So probably it's the DNA which is being transferred from here to here, converting the live R strain to what? S strain. You got it? But again, you needed experiments to prove that, which is why Finally, Hershey and Chase came up with their experiment. Okay, and they used bacteriophage and E. coli for their experiment. And they finally proved that DNA is the genetic material. So three different groups of scientists came. First was Griffith, second Avery, MacLeod, McCarthy, and then Hershey and Chase came into the picture. Now if at all you are asked to write the answer, you have to write the answer like this. Frederick Griffith conducted his experiment in 1928 using the bacterium this. There is 2 strains, one S strain, one R strain. S strain is virulent, has a polysaccharide capsule. This is non-virulent, lacks the capsule. Live S strain injected into the mice died. S strain is virulent. Live R strain injected into the mice, it lived because it is non-virulent. Heat kill S strain injected into the mice, dead bacteria cannot cause what? Any disease. So this was being found, and lastly they said the live R strain bacteria were transformed into virulent S strain. Griffith concluded that some heritable material, or you call it as transforming principle, transferred from the S strain to the R strain, but he was not able to find what was it. Cool? Okay, cool. Now we go ahead with the next question right up there, and this is from model question paper 3, and the question number is 5. So, okay, anyways. Match the following: Alec, Jeffrey, F. Sanger, Jacob and Monod and Griffith. You have to match the following. Alec Jeffrey was responsible for what? So the easiest thing that I know is that Griffith was the one who did the transforming principle experiment. So D goes with 2, and if at all I know that, then I can match this following, right? A goes with 4. Alec Jeffrey was the one who went ahead with DNA fingerprinting. F. Sanger, Frederick Sanger, my dear children, he was responsible for annotated DNA sequencing, the whole DNA sequencing. Okay, so S for Sanger and S for sequencing, you remember like that. Alec Jeffrey has an F over here, fingerprinting also has an F. Now, going ahead, Jacob and Monod, they gave the lac operon model. They gave the lac operon model, so C goes with 1. C goes with 1. So here goes your answer. It's an easy question. I hope these type of questions also show up in your exam so that you can easily grab marks. Okay, now just give me a second. Okay, so now the next question that we have is a 5-marker question. Enumerate the features of a genetic code. So these are the genetic code, my dear children, and what are the features? How many codons do you know, my dear children? We have almost 64 codons, out of which 61 codes for amino acids, and there are 3 of them that basically are stop codons. Okay, so the codon is triplet. Always remember they come in number of 3, 1, 2, 3, right? And 61 codons code for amino acids, and 3 of them are not coding for any amino acid. They stop the process of translation. You call them as stop codons. Now, the first codon which is there, which is super important for the process of translation to begin, for the process of RNA to protein to begin, you need a very important codon, and that is AUG. AUG has dual function. First, that it helps in initiating the process of translation, and also it codes for an amino acid that is known as methionine. So basically dual function because it is an initiator codon and it codes for methionine. Second thing, some amino acids are coded by more than one codon, hence the code is degenerate. That means, for example, if at all you see phenylalanine, or let's say leucine. Okay, leucine is coded by CUU, CUC, CUA, CUG. All of them are coding for the same amino acid. All of them are coding for the same amino acid, which is why you will call them as degenerate codons. Now, the codon is read in mRNA in a contiguous fashion. As in, for example, I have a strand of mRNA. You do not have any full stop, commas, anything out there. There are no punctuation. They are read continuously. Now, the 3 stop codons I spoke about, remember their names. What are their names, my dear children? UA, UGA, and UAG. UA, UGA, and UAG. These are the 3 stop codons. You also call them as terminator codons. Now, the codons basically are generally universal. For example, UAU. In our body, it codes for tyrosine. In a bacterium's body also, it codes for tyrosine itself. So you say it is universal. From bacteria to humans, UUU would code for phenylalanine. That's an example. Now there are some exceptions. Obviously, every time there are exceptions. So some exceptions to this rule have been found in mitochondrial codons. Okay, the codons found in mitochondria. In mitochondria also we have the DNA and things like that. Do you know that? Apart from nucleus, there is mitochondrial DNA. So there also transcription is happening. There also we have mitochondrial codons. And in some protozoans also, there are something which are very different from ours. Okay, so these are the things that you have to keep in mind. And also remember, codons are unambiguous. Unambiguous means one codon, for example, UUU, only one amino acid. It does not code for multiple amino acids. Okay, so children definitely gets confused between degenerate and unambiguous feature. Degenerate means multiple amino acids, or one amino acids can be coded by multiple codons are there. One simple amino acid can be coded by multiple codons. And always remember, one codon, only one amino acid, nothing else. That is unambiguous nature. So these are the features of genetic code. This is model question paper 3, question number 38. I paused the video, I made some changes here and there. I saw some questions were missing. That's why the number of questions would also be different. Okay, so do not be worried. I just included more questions, and by this I have added one more question right up over here. Where is that question, man? Okay. So the 5-marker question which was missing, from here is related to lac operon. This is model question paper 2. So we discussed one 5-marker question, and that is Griffith. But before that also you had a question. So the question number can be, okay, wait, just a second. Okay, so this is 39th question, and this would be 38th question in the case of model question paper 2. So we missed this question. It is dependent upon lac operon, but don't worry, we'll see a similar question right now in model question paper 3, and I'm so sorry. It's even a better question, so I can explain everything out there. Here they're just asking you what is I gene coding for, what is LAC-Z, what is Y, and what is A, what is inducer, all of them coding for. What is their function? There I'll explain everything about lac operon, so please do not be worried. Now if we talk about model question paper 3, the 2-marker question which is missing, actually they gave the same question. Okay, this question, same question is repeated there also. That's why I written model question paper 2 and 3, question number 24, it's the same. So it's this, and now we will go to what we were talking. I know it's too much of confusion, but I just wanted to come back and clarify things so that you get to understand that we have completed the questions. Okay, so yes, question number 10. State the arrangement of different genes of lactose operon in E. coli. Describe the role of lactose in regulation of lac operon. So this is the question I was talking about. So my dear children, you know how does it work? So the flow of genes is being asked, the arrangement of different genes. First of all, you have a very important gene which you call as regulatory gene. What do you call it? Regulatory gene. It regulates the whole process by producing, over here it is producing a repressor protein. Repressor protein. Now before any gene which is there, you will have obviously a promoter region for that gene. So there is promoter region for the regulatory gene which is denoted by I. Then now you have promoter gene as well as operator region for our main structural genes, that is Z, Y and A. This is what? Promoter. This is operator for the structural gene which is being regulated by the regulatory gene. If you have doubts, I know you might have, listen to me carefully. Z over here codes for beta-galactosidase. It's an enzyme which basically works on lactose, breaks it down into glucose and galactose. Okay, Y is, where is it? Y is another gene coding for an enzyme permease. Permease increases the permeability of the membrane. So if these genes are all inside the E. coli, my dear children, Escherichia coli, right? Now to increase the permeability of the membrane of E. coli, we have permease. It increases the permeability. Now we have A gene for coding for another enzyme, trans-acetylase. It basically, the function is clearly not given in NCERT. It basically works with the byproducts that you get throughout the process. It deals with all of it. Trans-acetylase. So my dear children, this is the arrangement of lac operon. Why do you think lac operon is arranged like this? Now listen to me. As I said, all of this is happening inside the E. coli. Now, E. coli can either depend on glucose or lactose for energy. Suppose in the medium where we are growing the E. coli, we have put lactose. You need to break down this lactose, right? First of all, you need to make this lactose inside. For that, you have permease enzyme. Then you need to break it down. For that, you have beta-glucosidase. And for everything in between, you have trans-acetylase as well, right? But whenever lactose is not there, do you think there is a need of any of these enzymes? No. That means they need not be produced. That means this whole thing, lac operon, my dear children, this is a lac operon, it should be switched off. It should be switched off. And how do you switch it off? So the repressor protein right over here, my dear children, goes and binds on the operator region. It goes and binds on the operator region. You know, who is responsible for transcribing? It is RNA polymerase. RNA polymerase always comes and binds on the promoter region wherever it wants to do transcription, and then it moves ahead for the process of transcription. But over here it is not able to move ahead. Why? Because there is a repressor protein being attached, and hence the enzymes won't be produced, and hence it's totally fine because lactose is not there. But suppose lactose is there in the medium. Suppose lactose is there in the medium. And suppose lactose has got inside the cell also. Now you need to switch it on. Now you cannot keep it switched off. You need to switch it on. So you need to remove the repressor protein. But how do you remove the repressor protein from here? Basically, lactose comes and binds with the repressor protein. Now it is not free to go and bind over there. So which is why transcription happens. Later on, the extra lactose which is there is being broken down. But how did the lactose enter at the first place if permease enzyme was not being made? Remember, even though if we say it is in switched off condition, little amount of enzyme is still being produced. Little amount of transcription is still being happening, which is why they are permeable enough to get inside. Now this lactose which is there, it acts like an inducer, because it has induced or basically started off the process of what do you call, or switched on the lac operon. So this is what? An inducer. Similarly, if lactose is not there, you have another thing which is known as allolactose. Which is known as allolactose. This allolactose which is there, my dear children, can also act like an inducer. So state the arrangement of different genes. It is like this: promoter, then I gene, then promoter, then operator, structural gene ZYA. Now, you have been asked that describe the role of lactose in regulation of lac operon. So while you mention that, please make sure it's a 3-marker question. The first part of the question is a 3-marker question. So make sure you write something a bit more about what are they doing. So I gene is a regulatory protein coding for repressor protein, promoter site where RNA polymerase binds, operator site where repressor binds to block the transcription, structural genes, this, what do they do? All of them has to be maintained right up over here. You show the picture as well, and then you talk about role of lactose in the regulation of lac operon. Now that is not 2 markers, that is actually, sorry, not 3 markers, it's a 2-marker question. So in the absence of lactose, the repressor protein binds to the operator. This blocks RNA polymerase. So structural genes are not transcribed. In the presence of lactose, what is going to happen? You have to tell that. That's all, that's the role. And now, if at all we go back to this question that I told you is a similar kind, you would be able to answer this. What does I gene do? It codes for repressor protein. It's a regulatory gene. It is coding for repressor protein. This repressor protein binds out there on the operator region, does not allow transcription to happen. All of it you have to write. LAC-Z gene codes for beta-galactosidase. Breaks down what? Lactose into glucose and galactose. Okay, Y gene codes for permease, increases the permeability of the cell wall so that the inducers such as lactose and allolactose can get inside. And LAC-A gene is trans-acetylase, detoxifies the cells and things like that. Inducer which is there, as I told, it binds with the repressor protein, does not allow the repressor protein to go bind on the operator region. So for all of them you need to explain and explain properly and you will get your whole marks. Make sure you do not forget to make the diagram. That is important. You need to make this diagram. Okay? Cool. I hope it works, and with that I think all of it is totally completed. Now am I missing on something? I hope no. Allolactose and lactose, please remember both of them are inducers. Okay, I was going to say something but somehow I forgot. Okay, now going into the revised question bank questions. Here you will be seeing more questions, so do not be worried. Let's go get into it. The first question that we have is GGU, GGC, GGA, GGG all code for glycine. Which salient feature of the genetic code does it prove? What feature does it prove, my dear children? Unambiguous nature, degenerate codons, universal code, initiator code. 4 amino acids coding for the same, 4 codons coding for the same amino acid. What features does it show? It is degenerate. Degenerate, multiple codons, one amino acid. So it is degenerate. If it was said that GGU codes for glycine, UUU codes for phenylalanine, what does it prove? It shows unambiguous nature, one codon, one amino acid. So here it is degenerate nature which has been said. If at all the question is, this codon codes for this in human beings and the same thing is being produced in bacteria, what feature does it show? Universal codon feature it shows. Okay, so it's the question bank question number 6th question. Now, in a single-stranded DNA, if the ratio of A and G is 60 and 40, then the ratio of T and C is. So all of a sudden when children see this question, what do they do is, if A and G is 60 and 40, then A will equal be T, right? So it will be also 60 and here also it will be 40. So 60 and 40, this also will be 60 and 40. My dear children, read the question carefully. It's not double-stranded DNA. They are asking about single-stranded DNA. In a single-stranded DNA, if the ratio of A and G is 60 and 40, then what's the ratio of T and C? That's the question. So it's in single stranded, how can you tell? Now A and G came, similarly you can have multiple T over here, multiple C over here. You cannot define that. It's a single stranded thing. We can only talk if it is double stranded. So none of the above would be your answer for this question. Okay, it's none of the above. Okay, children, you think that you are able to understand things better, or basically you are getting to know some extra questions which you were not expecting, do not forget to share it with your friends and do not forget to hit the subscribe button right now, because I'm telling you, this channel has helped so many children out there, so many children the previous year, and I can guarantee you we are still helping you and we will do it in the best possible way ever. Yes, so keep that trust on us, and yeah, that's all. So the components of deoxyguanosine is what? Whenever you have a term like that, please understand guanosine, adenosine, all of it means that it's a nucleoside. What do you mean by nucleoside? Sugar with a combination of nitrogenous base is a nucleoside. So what sugar? It can be deoxyribose sugar or ribose sugar. If it is DNA, then deoxyribose sugar. So deoxyguanosine is just guanine, that is a nitrogenous base, plus deoxyribose. It does not include the phosphate group. If at all you want to include the phosphate group, it should be deoxyguanosine monophosphate. At the end, it should be monophosphate. Here, until guanosine is only given, that means it's just sugar plus the nitrogenous base, nothing else. So your answer for this question becomes B. Okay? Next. A mature processed mRNA transcribed from a gene is 636 nucleotide long, assuming it codes continuously without UTR and stop codons for a polypeptide. How many amino acid does it encode? So usually what happens, my dear children, in an mRNA, in front, okay, before the 5' end, you have some untranslated region that is not going to get translated. And here also, before the 3' over here, my dear children, you have the stop codon which stops the process of translation. But here they are asking you, do not assume that there is UTR, do not assume there is stop codon. There are only codons which basically are coding for amino acid. So 636 nucleotides are there, my dear children. If you want to see how many codons are there, how do you do? So each of them basically have to be coming together in the form of 3 to form a codon. So 636 divided by 3 is equals to what? I have 212 codons. And what does this codons do, my dear children? They code for amino acid. They are going to code for amino acids. So 212 codons codes for 212 amino acids. You get it? So 636 is a number of nucleotides. You have to divide it by 3 because they are going to come in a number of 3. Now you got 212 amino acids, 212 codons. 212 codons codes for 212 amino acids as well. How many number of codons? That many number of amino acids. So your answer is 212. Next, statement 1. Deoxyribonucleotide triphosphate. Okay, dNTPs. What do you mean by that? Deoxyribonucleotide triphosphate. That means basically over here you have your deoxyribose sugar, you have your nitrogenous base, you also have phosphate, not just one phosphate. How many phosphate? 3 phosphate. You call this as deoxyribonucleotide triphosphate. Now they act as substrate during DNA replication. Do they act like substrate? Substrate is something that with the help of which we can form something else. So during DNA replication, what is happening? Suppose here is a strand with A, T, G, C. Now on the opposite strand, my dear children, we need these things. We need nucleotides. And how do we get nucleotides? In the form of this. So they are actually the substrates. Now when they come together, what will happen, my dear children? When they come together, basically this bond is broken. So there is only one phosphate left, rest 2 phosphates are gone. And when they are gone, they release basically so much amount of energy. When this bond is being broken, they release so much amount of energy. This energy is something that helps in the formation of bond between multiple nucleotides. So they are the substrates. And they also are the energy givers. They are the substrate, they also are the energy givers. With this energy, the bonds are being formed over here between each nucleotides. So deoxyribonucleotide triphosphate, dNTPs, acts as substrate during DNA replication. Statement 2, deoxyribonucleotide triphosphate also act as source of energy in the process of DNA replication. Both the statements are correct. Both statement 1 and 2 would be what? Correct. Got it? Yes, ma'am. Next, in pea plants, each character is governed by a gene which is in pea plants. So my dear children, Escherichia coli, we saw that a structural gene structural gene is made up of Z, Y, A. So there is multiple genes. If I put into 3 things, there is gene 1, gene 2 and gene 3. But all of them are together. But all of them are being together having a common promoter and everything. You call this as polycistronic condition. You call this condition as polycistronic condition, where multiple proteins are going to be made or multiple genes are being located in the same structural unit. This is known as polycistronic condition. This is found in prokaryotes. So P for polycistronic and P for prokaryotes. Whereas in us, in eukaryotes, my dear children, one structural gene means one gene only. This gene is going to code for one protein. That's all. Only one gene. One promoter for one gene. That's how it works. So this condition is known as monocistronic. Now, ma'am, why are you talking about us? It has been asked about pea plant. Plant are what? Plants are also eukaryotes. All eukaryotes, my dear children, are monocistronic. Prokaryotes are polycistronic. So over here, your answer would be monocistron. In pea plant, each character is governed by a gene which is monocistron. Okay, there is no polycistronic thing in the case of us or eukaryotes in general. Now, which of these are correct? UTR, I just told you, if you talk about an mRNA, you have 5' end, you have 3' end. Okay, this is the region that is going to get translated. Before the 5' end, you have UTR, untranslated region. After the 3' end also, you have UTR, untranslated region. Why do you think it's important? To make the mRNA stable, to make it stable, not just that, for process of translation to begin, for initiation of process, you do not just need initiator codon, you also need UTR. So for initiation to happen of translation, for making it stable, for protecting it, for efficient translation to take place, we need UTR. We need untranslated regions. So UTR, untranslated regions, are located on the DNA is the first statement. Untranslated regions are located on the DNA. Is it correct? Let's go. UTR is present both at the 5' before the start codon and also at the 3' after the stop codon. Correct? This is correct. UTR provides stability to mRNA. Correct. UTR is required for efficient translation. Correct. What is not correct? This is not correct. This statement would not be correct. So what is correct? 2, 3 and 4. 2, 3 and 4 would be correct. Hence your answer is D. Your answer would be D. Okay? Okay. Let's go ahead. Next, in a DNA strand, if there are 100 nucleotides and 30 are adenine, the number of thymine, guanine, cytosine respectively are. In a DNA strand, if there are 100 nucleotides, okay, total number of nucleotides is 100. Now, 30 are adenine. Adenine is equals to 30. The number of thymine and guanine, cytosine, this is double-stranded DNA you're talking about, okay? You're asked about guanine, thymine and cytosine. Listen, if it's a double-stranded DNA, how many number of adenine is there, that many number of thymine will also be there. 30 plus 30, you have got 60. So out of 100, 60 are them only. Out of 100, 60 are what? Adenine as well as thymine. So minus it, you have got 40. Now 40 will be guanine and cytosine. So do you do 40, 40? If you do 40, 40, what will happen? 30 plus 30, 60, then 40, 100, then 140 will be there. You have to divide this 40 into 2. So if this is 20, this will also be 20. Now you add it up, it will come out to be 100. 50 plus 50 is equals to 100. So how many number of thymine is there? You go in order. Thymine is 30. Thymine is 30. There are two options like that. Guanine is 20. Guanine is 20. I have two options like that. Cytosine is also 20, but here it's wrong, so your answer would become A. It becomes A. Cool? Yes, ma'am. Okay, next. In a structural gene consisting of 300 base pairs, if 4 nucleotides are added and 1 is deleted at the end point, it leads to. Any idea? If in a structural gene consisting of 300 base pairs, if 4 nucleotides are added, okay, 4 nucleotides are being added. Suppose it is A, U, G, C, and 1 is deleted at the end point, and 1 which is there, it is being deleted at the end point. It leads to what? Point mutation, frame shift mutation, unaltered change, 101 amino acids. This is a homework question for you guys. Try to solve it and let me know. This is confusing. This is a homework question. You have to solve it and let me know. Okay, and if you face any trouble, let me know that as well. I'll solve it for you. But this you should be trying. This question I really liked, and yes, as I said, you should be trying. So I'm not showing you the answers right up over here. You try to solve it. Now we will move on to the 2-marker, the 5-marker questions which are usually have come. Okay, in the previous year model question paper and things like that. So which sequence of bases transcribed from DNA are found both in hnRNA as well as mRNA? Which sequence of bases transcribed from DNA? So you have the DNA right over here. They are asking you, the DNA is getting transcribed to what it is known as hnRNA. And this hnRNA undergoes processing to form what? The mRNA. So they are asking you what sequences are there transcribed from DNA which are found both over here as well as here. So my dear children, I told you that in our DNA we have the coding region, you call it exons, and the non-coding regions you call it as introns. This is how it goes. The same thing is transcribed up over here, everything is same. hnRNA, my dear children, undergoes processing. undergoes processing. In processing, there is a process known as splicing, where the introns are removed. The non-coding regions are being removed. Now what are you left with? You are just left with exons. Not over here, over here. You are just left with exons. Now tell me what's common over here and here. What's common over here and here? It's just the exons which are common. It's just the exons which would be common. So the sequence of bases transcribed from DNA that are found both in hnRNA and mRNA are what? Exons. Exons are the coding regions that remain in the mRNA after splicing, while introns, not coding regions, are removed during what? The process of processing. I hope it's clear. Apart from splicing, there are a few more steps such as capping and tailing. Capping and tailing. So here, guanosine residues being added over here. At the end, you have so many adenine residues, polyadenylation you call it, that is known as tailing. That all is being done on the mRNA, on the hnRNA. Finally, you get the mRNA, and then this mRNA will be moving out of the nucleus. After the processing is being done, it moves out of the nucleus. Why do you think this capping and tailing is being done? So that it can be protected from any enzymes once it goes outside. Okay, that is why it is being done. So it is super important. Please remember that. Please go through the details of all these processes. Questions can come anytime from anywhere. So I am trying my best to cover as much as possible, but still I am telling you, please make sure wherever you have any doubts coming in, okay, what is capping? Let's see. Go and check it up. What is tailing? Let's see. Go and check it up. And these can help you. For example, you're writing some answer, you can get additional information also there, and you might get some good bonus marks. Okay. So please do that. Now question number 21, mention the levels where gene expression can be regulated in eukaryotes. So my dear children, I told you in prokaryotes, everything happens up over here in the cytoplasm. Everything. Okay. Transcription, ma'am, here. Translation, ma'am, here. So transcription and translation happens coupled. Okay, it's a coupled process. So that means transcription is happening and then translation is happening. It's happening simultaneously, my dear children, in the case of prokaryotes. But in the case of eukaryotes, things are quite different. In the case of eukaryotes, we have a nucleus. The DNA gets transcribed over here in the nucleus. So I can make some changes over here such that this transcription does not happen. If I do not want the proteins to be made or the genes to be expressed, first thing, at the transcription level, I can make some changes. Now suppose the transcript is made, okay, after that, after the transcript is made, that is hnRNA is made, you also need to undergo processing. I can make sure that there is no processing happening. If I do not want the gene to express, I can make sure that there is no processing happening. So at processing level also I can control. Now my dear children, this mRNA which is being formed after processing, suppose it is being made, I can make sure that it does not go out. Transport of mRNA from the nucleus can be stopped. In that level I can regulate. Now, after coming out, my dear children, in the cytoplasm, this mRNA can undergo the process of translation as well, and I can stop that also. This is the 4 ways where I can control or I can check the regulation of gene expression in the case of eukaryotes. But in the case of prokaryotes, it's not like that. Okay, everything happens over here itself. So for them, regulation happens with the help of operators and things like that, as you saw in lac operon. Okay, so always remember, in them, transcription, translation is a coupled process. But here, it's not like that. It cannot happen simultaneously. Now, write a note on the role of sigma factor and the rho factor in transcription of prokaryotes. Write the role of sigma and the rho factor in transcription of prokaryotes. So, my dear children, what exactly happens is, I told you, the DNA which is there, okay, it is transcribed with the help of RNA polymerase. Now, RNA polymerase goes and binds on the promoter region of the DNA. So, there is a region that is known as promoter region, but it cannot do it alone. It cannot initiate the process of transcription alone. It needs a factor, you call it as sigma factor. Now, once transcription has started off, it starts crying. Towards the end, it starts crying. It's so sad to leave the DNA, my dear children. So, while it is crying, you need a rho factor. Rho as in Rona in Hindi, that's why I said rho factor. So, to end the process of transcription, when it reaches the terminator region, you have an addition of thing that is rho factor. When rho factor comes, then only you can end up the process of transcription. So you have sigma and the rho factor. Sigma factor binds to the RNA polymerase, guiding it to the promoter region for transcription initiation. It detaches after initiation. Rho factor facilitates termination by binding to the RNA and unwinding the RNA-DNA hybrid in rho-dependent termination. It makes sure that the RNA-DNA is being separated. Now, one very important thing that I had to mention over here is that in your NCERT it has given that in the case of prokaryotes, okay, in the case of prokaryotes, you do not need multiple RNA polymerase. You just have one type of RNA polymerase. That's all. Whereas in the case of eukaryotes, we have three: RNA pol 1, RNA pol 2 and RNA pol 3. You mention it is in Roman numbers as in 1, 2 and 3. One is responsible for formation of rRNA, ribosomal RNA. Have you heard about ribosome? Have you heard about ribosome? It's an organelle. So this organelle, my dear children, is made up of rRNA and some proteins. So ribosomal RNA is the first thing. Okay, RNA polymerase 1 helps in the formation of ribosomal RNA. RNA polymerase 2 is the one that helps in transcription. That means formation of hnRNA, mRNA in the case of what? Eukaryotes. RNA polymerase 3, which is there, is responsible for the production of tRNA or transcription of tRNA. tRNA is transfer RNA. So 3 RNA polymerase 3 for tRNA. TT, you can remember like that. 2 for hnRNA, that is the secondary function. RNA or ribosomal RNA is the primary thing that it has to make. So rRNA is the one made by RNA polymerase 1. Now apart from tRNA, there is 5srRNA. Okay, 5srRNA, that is a different type of rRNA which is coded by third one, okay, RNA polymerase 3. So please go through all of that once. It's there in your NCERT, just giving you hints on what all type of questions can be coming in. Okay. Now, write the difference between euchromatin and heterochromatin. Name the virus used in Hershey and Chase experiment. Okay, two different questions. So, write the difference between euchromatin and heterochromatin. My dear children, I told you there is nucleosomes. When the nucleosomes, suppose they are far away from each other. Okay, so basically you have area, this is a DNA, right? You have area for the RNA polymerase to come, bind and do the process of transcription. So I'll say this type of chromatin is transcriptionally active. Transcriptionally active. What do you call this type of chromatin which is loosely packed? You call this as euchromatin. It is loosely packed and it is transcriptionally active. It is loosely packed and transcriptionally active. Now, my dear children, if at all children, if we go ahead and it is stained, okay, it stains, but it lightly stains. It stains light. Why, my dear children? Because as you can see over here, they are loosely packed, which is why the stain also is taken up lightly. Now you have one more where the chromatin is very densely packed. What do you call this as? This is known as heterochromatin. Heterochromatin. And my dear children, it is tightly packed. It is tightly packed. It is transcriptionally inactive because there is no space for the RNA polymerase to come in. So it is transcriptionally inactive, I've written over here, and it stains very much dark. It stains dark. So in a typical nucleus, some loosely coiled regions of chromatin with light stain and transcriptionally active DNA is known as euchromatin. Whereas the chromatin that are more densely packed and stains dark and transcriptionally inactive is what? Heterochromatin. Cool. So this is the heterochromatin and this is the euchromatin region. Now name the virus used in Hershey and Chase experiment. Ma'am, virus, huh? You just use bacteria, that is E. coli, and bacteriophage, no? The bacteriophage which is there, it is a virus. Okay, so the answer for this question is bacteriophage. It is bacteriophage. Now, list the goals of Human Genome Project. What are the goals of Human Genome Project? To identify all the genes in the human genome. And after identification, my dear children, after identification, what do you have to do? Basically, you have to store the information. But before that, first of all, to identify all the genes, and for that, we need to determine the complete sequence. We have to determine the complete sequence of the DNA, and after that, identify the genes, all genes, what are their functions and everything. Then, to store information in the database, and then improve the tools for data analysis. You stored it, but you need to analyze also. So you need to improve the tools for data analysis. Next, transfer related information to other sectors and industries. Transfer related information to what? Other sectors and industries. You cannot keep it for yourself. You have to help the other industries also, and address the ethical, legal and the social issues arising from the genome studies. So all of them, my dear children, have to be cleared up. Okay, identify all the genes in human genome, this, all of it. If you write, you get your marks. Now, given below sequences of nucleotides in a particular mRNA and amino acids coded by it. So, AG, methionine, UU, phenylalanine. Write the properties of genetic code that can be correlated from the above given data. So, you have been asked, this is the thing, you have to tell the genetic codes feature. So, we already discussed it. This came in the previous year model question paper and also July 2022. So, over here, AUG is the initiator codon. So, initiator codon has dual function to initiate and to code for methionine. You see both of them codes for phenylalanine. That means it is a degenerate codon out there. GAG is coding for glutamine and UU is coding for leucine. All of them show unambiguous nature of the codons. One codon, one amino acid, right? UAA does not code for anything. That shows that it is what? A stop codon and or a terminator codon. Apart from that, there is no comma, there is no full stop, it is contiguous. So all the features have been described, you can talk about it. So I have mentioned all the features, but yes, you can pick whatever I have just told and write it down. But it's a 5-mark question, it will be very, very safe if you write everything. Cool? Cool, ma'am. Now, describe the steps involved in DNA fingerprinting process. What are the steps which are involved in DNA fingerprinting process, my dear children? Any idea? What is DNA fingerprinting? So, our DNA which is there, it is very unique. None of us share the same type of DNA. Okay? And my dear children, in our DNA, we have some regions which are known as, we have some sequences which are known as variable number of tandem repeats. These sequences, my dear children, for example, in my body it is AGCT, which is repeated 2 times. In yours, the same thing might be repeated 4 times. In someone else, 6 times. Similarly, we have what? Differences with that. Also, we have some, sequences which are very unique to us, not found in anyone else. We get it? So, with that principle, we are going to do some experiment, and that is known as DNA fingerprint. It's not an experiment, it's a process, a technique. Usually used to find the criminals, or if at all there is some disputes within, like who is the father, mother and things like that. At that particular time this is being used. And always remember, where is our DNA being found? If at all you need samples, you can have either saliva or your hair, all of it, saliva, hair, all of it, not the blood, not the blood cells. Blood cells, for example, RBC, if at all you take RBC does not have a nucleus. In nucleus is not there, DNA will also be not there. So please understand the small details out there. So my dear children, someone, X person did a crime, and this is X person's DNA. Now I sequence the whole DNA. And got to understand that I can make a complementary DNA strand to it. I made a complementary DNA strand to it, and basically I tagged it with something. Okay, I tagged it with something which will show up when I put it under UV radiation. So this strand of DNA which is there, I call this as a probe. I'll need it later on. So this is the thing that I found out from the crime scene. So suppose there are 2 people whom I am confused with. It is A and B. A is me and B is you. And my dear children, what happened was, basically our DNA was taken, both of our DNA was taken. The DNA was being cut using restriction enzymes. Okay, the DNA is being segmented into parts using restriction enzyme. That is the first thing. So first is isolation. After that, cutting using restriction enzyme. Then you do gel electrophoresis. Then you do what? Gel electrophoresis. That means you make the cut DNA run through a chamber. So when you run through a chamber, my DNA and your DNA, it looks like this, okay? My DNA strand, it goes like this. And yours, it is in gel electrophoresis chamber. Now I cannot see it. For that, basically, what do? I transfer the DNA which is there on a nitrocellulose sheet. This is known as southern blotting. This is known as southern blotting. Basically, transfer of DNA into a nitrocellulose sheet. Now on this sheet, my dear children, I am going to put this particular probe. I am going to put this particular probe. Now the probe which is there, it will only go and bind in the complementary region. So suppose it went up over here and it bound it there. You have multiple probes being made. Okay, you have multiple probes being made and they are binding up over here. Similarly, the probe is binding up over here also, somewhere over here. Now, this DNA which I got, that also I am going to make it undergo cutting by restriction enzyme and then electrophoresis. And basically, I also have a pattern similar to this, where when I am comparing, I get to see that wherever the probe attached over here, the same places it is getting lighten up over here in the suspect. Whereas the probe is attaching over here, but at a very different place. We all have some common factors, which is why it is attaching up there also. Complementary pairs are found there also. But this is exactly the same. And with this, my dear children, I get to understand that the suspect is not anyone else but me. I did the crime. This is DNA fingerprinting method. And how do you put it in the answer? Isolation of DNA. DNA is extracted from the sample. It can be blood, hair, saliva, etc. But in blood, not the RBC. You have to make sure of that. So please cut it out. Okay, now digestion by restriction enzymes. DNA is cut. Blood as in blood you can take, but not erythrocytes. Okay, not the erythrocytes. Please remember, in the blood, you don't just have RBCs, you have multiple things, but erythrocytes or RBCs cannot be referred because it does not have a nucleus. If erythrocytes have been mentioned, please understand they do not have a nucleus. Anyways, digestion by restriction enzyme. DNA is cut into fragments using specific restriction enzymes. Separation by gel electrophoresis. The DNA fragments are separated based on size using agarose gel electrophoresis. Transfer to membrane. The separated fragments are transferred onto a nylon or nitrocellulose membrane. Hybridization with DNA probe. Radioactive. So either I can attach the radioactive thing with the DNA out there or any other thing, okay, fluorescent thing. So radioactive or fluorescent probes complementary to the specific sequence bind to the variable regions of the DNA. Visualization. The hybridized fragments are visualized using X-rays or UV light, producing a unique DNA pattern. This pattern is compared for identification or to identify the relationships. And suppose you have the same thing, is the same. Suppose mother and daughter would be having so much similarity rather than someone else. You get it? So in that case also it can be helped. Fine. So I'll suggest start off with DNA fingerprinting, what you call a little about DNA fingerprinting, why is this used, for example, as I said, for finding, for solving any criminal cases or for finding relationship where maternity and paternity disputes are there. Just have a quick intro about it and then go ahead with these steps. Now, question number 27. Answer the following. Construct a complete transcription unit with promoter and terminator on the basis of hypothetical template strand given below. So this is the hypothetical template strand which is given to you, my dear children. Okay, this is the template strand given to you. You have to create what? A transcription unit. Write the RNA strand transcribed from the above transcription unit along with the polarity. And you have to also tell what is the RNA which you can get transcribed after getting transcription. So over here, this was the sequence. Okay, you need to understand that this is the 3' end and this will be the 5'. Arrow will be in the 3' direction. So 5' and 3'. Okay. Now, my dear children, usually what happens, I'll tell you. So this is 3, this is 5, this is 5, this is 3. Promoter region, okay, this is the strand which is going to get transcribed, you call this as a template strand. Why? Because it is in 3' to 5' direction. Now the RNA polymerase binds over here and starts off RNA polymerization in 5' to 3' direction. Whereas the other strand which is not going to undergo transcription, you call this as coding strand. Now where is the promoter region located? Where is the promoter region located? So over here, towards the 5' end of the coding strand, we refer like that only, promoter region will be located. Towards the 3' end of the coding strand, the terminator region will be located. That is how you usually say. So this is your 5' to 3', so this part is going up over here. This is going to go up over here. That means A, T, G, C, A, T, G, C, and blah, 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 blah. So over here you have to make the coding strand. Opposite of it, A goes with T. Just a second. Yeah, A goes with T, T goes with A, G goes with C, C goes with G, A goes with T, T goes with A, C goes, sorry, it was G, right? J goes with C and C goes with G, and so on. This is your coding strand, this is your template strand, you have to write everything. Now they are asking you one more thing. Tell the RNA strand which is going to be made from this. So how do you say? So opposite to this, you have to make an RNA strand. You have already made an opposite DNA strand. You just have to do one change. That is what? Wherever T is there, replace it by U. Wherever T is there, replace it by U, because in RNA you do not have thymine, you have uracil. So it will be 5' UACGUA. Wherever T is there, replace by that, and that's all. Here is your answer. So you do not make like this. Rather, my dear children, put the promoter region like this. Okay, make the whole promoter region like this. You can extend it 5'. This is your promoter region, and here also terminator region, not like that. Put it like this, put terminator region, and you extend it. Okay, both the strands should be covered. Cool? Cool, ma'am. So you can make it like this, properly write everything out there. I have not mentioned. Make sure you're putting the 3' and 5' and 5' and 3', and mention that RNA polymerase works from 5' to 3', everything. Okay. List the salient features of human genome. This was asked so many times, also in the model question paper the previous year. What are the salient features? From this also you get multiple MCQ questions. Salient features. The human genome contains how many base pairs? 3164.7 million base pairs. How many? 3164.7 million base pairs. Everything is million over here. Now, the average gene, a gene which is there, usually it is, average gene is of 3000 bases, but sizes varies, with the largest known human gene being dystrophin at 2.4 million bases. How many? 2.4 million bases would be there in dystrophin. This is an exception. Usually gene size is just 3000, but over here it is made up of 2.4 million bases. Our whole genome is 3164.7 million. Out of that, if dystrophin gene is there, it is 2.4 million bases. Now, total number of genes is estimated to be 30,000 only, much lower than the previous estimates of 80,000 to 1,40,000. Almost all 99.9% nucleotide bases are exactly same in all people. We just have that 1% exception. Now, the functions are unknown for over 50% of the discovered gene. It is unknown for what? Over 50% of the discovered gene, and less than 2% of the genome codes for protein. Less than 2% of the genome is actually going to code for any protein out there. Now, repeated sequence makes up very large portions, and that is the reason I told you that only 2% is coding for protein. Rest everything is repeated sequence. They're just there. That's all. Repeated sequence are stretches of DNA sequence that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. So until now, this number 30,000 gene, only 50%, only functions of 50% of things we know. Okay, less than 2% basically actually coding for proteins also. Rest of them are repeated sequences. Now, MCQ question. Chromosome number 1 has the most genes. 2968. Here 7 is missing, all the other digits are there. 2968. Remember like that. Chromosome number 1 has the highest number of genes. Chromosome number Y, okay, which is the last one, has the fewest. It is found in the males. It is just 231 genes. This is a question. This is a MCQ question. 231 genes in chromosome number Y. Scientists have identified about 1.4 million locations where single base DNA differences, single nucleotide polymorphism, SNPs, occur in humans. How many locations? 1.4 million locations have been found where we only differ by what? Single base differences, single nucleotide differences, or you call it single nucleotide polymorphism. So 2968, 231, 2.4 million bases dystrophin, 30,000 genes, and 3164.7 million base pairs. These are the numbers you need to remember apart from the text. Why? Because it can come in the form of MCQ question as well. The largest gene is for dystrophin. It's a protein. Now, explain the process of translation in protein synthesis. What is the process of translation all about? So, my dear children, translation means what? You have your mRNA and basically you need to make protein out of it, right? Who is going to help you with that? You need few things. First one is the ribosome. In the ribosome, you have larger subunit and you have smaller subunit. Apart from that, you have tRNA. So tRNA is a structure that looks like this, 5' end, 3' end. Okay, here you have an anti-codon loop. So whatever codon is there in the mRNA, opposite to it, you will be having an anti-codon over here in the anti-codon loop. At the 3' end, you will be having an amino acid being attached. This process is known as amino acylation, or you also call it as charging of tRNA. Amino acylation or charging of tRNA, and it requires a lot of ATP, highly energy requiring process. It is happening at the 3' end, do not forget it. So we need all this. Here, the first thing that happens is amino acylation. This process is already being done, or charging of tRNA is being done. We now have what? Charged tRNA. So the mRNA which is there, it does not directly go and bind with the tRNA. Rather, even though charging of tRNA has taken place, it is ready. tRNA is there in the cytoplasm with the amino acid. The first thing that will be happening is the smaller subunit of mRNA is going to come and bind. Okay, then, so I'll technically write that this is the second step. First step is amino acylation. First step is amino acylation. Over here, the smaller subunit will come and bind on the mRNA. Now the tRNA will be coming and binding. Which tRNA? Initiator tRNA. Initiator tRNA will be now coming and binding up over here. This would be the third step then. Now, my dear children, the largest subunit would be assembling. The largest subunit will be now assembling, and you have a whole different complex now being formed. RNA is made up of rRNA, as I told you, and apart from that, multiple proteins. Here we have certain enzymes also. What do? So basically the ribosomes are going to move on the mRNA like this. And when they move, what exactly happens is that, suppose over here, as you can see, there is a tRNA with so many amino acids. Because the amino acids, so many tRNA came and they went. When someone goes, my dear children, they leave their amino acid and they go as it went. So over here, now you have another amino acid coming. This and this amino acid, they form a bond. Now they are bonded. This guy tRNA goes. You get it? The same thing is going to be repeated. Every time the ribosome is going to move, another tRNA going to come, another tRNA going to come, another tRNA going to come. There is bond being formed between the amino acids out there. The peptide bond that is being formed. So, translation is the process of synthesizing a polypeptide protein from an mRNA template. This process of translation takes place in the cytoplasm of a cell where ribosome, which you also call as protein factories, is located. First thing that happens is what? Activation or charging of tRNA, amino acid. So charging not of an amino acid but the tRNA. So over here, amino acylation of tRNA or tRNA charging is the process of attaching an amino acid to this corresponding tRNA. This is also called as charging of tRNA. Now initiation, mRNA binds to the small ribosomal subunit. The start codon AUG is recognized by the initiated tRNA carrying methionine. The large subunit comes together forming an initiation complex. Now, tRNA brings amino acid to the ribosome, matching codons on the mRNA through its anticodon. Peptide bonds form between the adjacent amino acid. catalyzed by peptidyl transferase. It's an enzyme. Peptidyl transferase catalyzes the whole process. The ribozyme which is there, okay, the ribosome which is there, it is also having some RNA, my dear children. As I told you, it is made up of rRNA and all, which acts like catalyst, which helps in increasing the pace of this particular process. So ribosome also act like a catalyst. You might have heard about 23S rRNA, 23 subunit R, not Swedberg's unit as in 23S rRNA in bacteria is the enzyme ribozyme. Is the enzyme what? Ribozyme. It is a catalyst helping in increasing the pace of the process of formation of bonds. Okay. So please remember, peptide bonds is being formed. For that you have peptidyl transferase. Apart from that, the rRNA which is there, it also acts like enzymes, ribozymes. Now ribosome move along the mRNA in 5' to 3' direction. Termination: when a stop codon UAA, UAG, UGA is reached, translation stop as the release factors bind to the codon, freeing the newly synthesized polypeptide. So the tRNA does not reach the stop codons, rather release factors, my dear children, would be coming up there. It binds to the stop codons and stop the process of translation. So this is the whole thing. Okay, this is the process of translation. Now, DNA replication is said to be semi-conservative. Why? Describe the experimental proof of Michelson and Stahl to show DNA replication is semi-conservative. Why is DNA replication semi-conservative, my dear children? So over here, if at all you see, okay, this is the parental strand. Now, when the DNA replication happens, the strands basically, they move apart from each other. A new strand is being formed. A new strand is being formed. Now, one is still there. One older strand and one new strand is being formed. So which is why we say the parental strand is still there, but only one. So this is semi-conservative. Not both of them are coming here. Only one. So this is semi-conservative mode of replication. And Michelson and Stahl went ahead to explain this. So it was told by Crick and Watson that yes, this is happening. But later on, my dear children, Michelson and Stahl went ahead with the explanation. So what did they do is that E. coli basically which was there, it was in a culture medium. It was grown in a culture medium having N15 isotope of nitrogen. Okay, it was grown in a culture medium having N15 isotope of nitrogen. It was NH4Cl, in that nitrogen was of N15. Okay. Now over here, when the E. coli's DNA, everything was centrifuged, it was being found that they were settling down somewhere over here. So in short, I know it's kind of confusing. Listen to me carefully, okay? So I grew the E. coli in a medium having N15. Now the DNA which will be there in the E. coli will be taking up this N15 because nitrogen is a component of the DNA, right? So it will be made up of what? N15 isotope, right? And now suppose I am centrifuging this DNA, I am able to find, and where I am centrifuging? In cesium chloride medium. So I am able to find that all of them are settling somewhere over here because it's forming a heavy chain out there. It's a heavy form of thing. So N15, it's a heavy band which is being formed over here. Now, the same E. coli I am going to grow, okay, this E. coli which were there, I am going to grow in a medium where it has N14 isotope. Where it has N14 isotope. And when I do that, okay, when I do that, what exactly happens is that one strand is this, another strand is this, the new strand which is being formed, it looks like this. So this is N14, this is also N14. Now when I centrifuge it, my dear children, what I am able to find is that in the centrifugation tube, I am not able to find the heavy band. But there is a middle band right over here. There is a middle band right over here, which is hybrid band. Because over here, one heavy chain and one light chain is there. One heavy chain and one light chain is there. One is of N15 and another is of N14. Now again, if I am growing 20 minutes over, for each E. coli division, you need 20 minutes. So first 20 minutes, I got something like this. Again for 20 minutes, that means a total of 40 minutes, I'll be having something like this. Why? Because my dear children, listen. This strand divides into two, this and this. So one is this, one is this. Now the new strand is always going to be green. Why? It is N14. Okay, it shows N14. So over here, I got a whole new thing where both the strands are N14. And over here, I have got one N14 and one N15. Here also one N14, N15. Here I have got a whole new strand. Why? Because these strands differentiated, but the new strand which was there is going to be made of N14 itself. So now again when you do the centrifugation thing, you find hybrid bands and also the light bands, which shows that basically DNA replication is semi-conservative. Otherwise you would not find multiple bands out there. You will find only one type of thing. You are finding multiple bands because of the same reason. So this was the experiment which was done, and a question is there. Okay, an extra thing which is there. Very similar experiments involving use of radioactive thymidine was also being done. Okay, to detect distribution of newly synthesized DNA in the chromosome was performed in Vishya faba, that is faba beans, by Taylor and colleagues in 1958. So in the year 1958, similar kind of experiment was being done, okay, using radioactive thymidine, and it was performed in Vishya faba plants. Please remember, it was done by Taylor and colleagues. This is again an MCQ question that can be coming up. So these are the things that I wanted to discuss. Apart from that, the concepts I want you to go through is, please go through the genomic size of all the organism. For us is 6.6. Okay, diploid content in our body is 6.6 into 10 to the power 9 base pairs. Okay, now over here haploid will be half of it. That is the one found in gametes. It will be 3.3 into 10 to the power 9. Please know that. What happens in Escherichia coli? What is its genomic size? All of it is also important. Please go through that. Super important. Apart from that, I think, yeah. So if there are more questions, we will be definitely solving it later on. But yes, for now, these are the very, very important questions that you need to do. Cool. So that's all. Thank you so much, my dear children. And if you have any doubts which are there please put it down in the comment section. I am here to solve them all. Okay. Wishing you the best, and thank you. Take care of yourself. Bye-bye.